right. Hi, everyone. I want to start by thanking the program committee for giving me an opportunity to tell you a little bit about our work. And I'd also like to thank the organizing committee for doing such a wonderful job arranging this conference in this magnificent city. So what I'm going to tell you about today is a method we've developed for doing multiple sequence alignments based on protein language models. And this is really the work of Claire Mcwhite, who is a very talented and creative researcher at Princeton University. Unfortunately, Claire couldn't come. She was too busy traveling for interviews, and so I'm giving the talk instead. So obtaining multiple sequence alignments is this really core problem in computational biology. It's one of the first things we really learn about. So on the left on this slide is a multiple sequence alignment just taken from Wikipedia and made by Clustel X. And it's such a central problem because it's a first step for many downstream analyses, including, for example, phylogenetic reconstruction, figuring out what parts of proteins are under selection or conserved. If you're trying to predict the impact of mutations, whether they're pathogenic or not, multiple sequence alignments lead to these conservation measures, which are so critical. If you want to predict structure, multiple sequence alignments are critical. If you want to figure out what the domains are within a protein sequence, again, multiple sequence alignments are critical. So since we're computer scientists, I'll just remind you what the overall formulation is of a multiple sequence alignment, and we're talking about global multiple sequence alignments here. So the overall problem is you're given a set of sequences, S1 through Sn, with lengths L1 through Ln, and a valid multiple sequence alignment inserts any number of gaps into each of these sequences such that the modified sequences, S'1 through S'N, all have the same length L, with this restriction that there is no I such that all the sequences have a gap in the ith position, and this ith position would correspond to a column in the multiple sequence alignment that you build. Of course, there are many possible ways to do this, so many possible multiple sequence alignments that you could have for n sequences. Which one should an algorithm output? So first, let's just talk about the biological goal. And the biological goal would be, given a set of protein sequences, each column in the MSA, or multiple sequence alignment, corresponds to amino acids that evolved from a shared ancestral amino acid sequence. Now we can talk about the computational goal. So the way we typically think about the computational goal is if we're given a method to score each column in an alignment, we want to find the multiple sequence alignment which has the highest score across all columns. And typically, the scoring scheme uses a substitution matrix, which represents the similarity between amino acids, which are empirically estimated by looking at lots of aligned sequences. And so you have an example of a substitution matrix up there. And then you also have some kind of penalty. You pay for gaps. And then you say maybe you're going to score each column using a sum of pairs measurement, a sum of pairs score. So traditionally, algorithms for computing multiple sequence alignments have worked as follows. So of course, we know that you can use dynamic programming to optimally find the best scoring multiple sequence alignment in kind of the regime that I just set out. But in practice, when you have multiple sequences, people tend to use these guide tree-based heuristic approaches where you build pairwise alignments and solve those optimally. And then you have a guide tree that tells you how to merge these pairwise and larger alignments into a bigger alignment. And this has been the paradigm for the last 30 to 35 years in the area of multiple sequence alignments. So of course, multiple sequence alignment methods work quite well, but they struggle in the twilight zone of sequence similarity. So this is the case where the structure and function is conserved, but sequence is not. So I want to say that MSA is an old problem. As I said, people have been working on it for more than 30 years. They've used these so-called progressive alignments, and really in the last decade or so focused on really scaling up these type of methods, including also making these methods work better. But even though it's an old problem, it's not a solved problem, and so there's room for brand new ideas. So everyone knows that there's been this revolution going on in natural language processing, and protein sequences also are like a language. So you can think of a protein as a sequence of tokens, where each token, in this case it's an amino acid, has a meaning that depends on the context. So in language, you might have, I had sea bass for dinner, where you know it's bass is a fish and not base the instrument, and you know that because of the context of the sequence. Similarly, you have your protein sequence, where each token is an amino acid, and you'd like to look at the histidine there and say something about the meaning of that histidine based on the context around it. In language models, each token is represented by a high-dimensional vector that captures its identity and context. And that's true in the case of natural language, with the little vectors below, as well as in the protein languages. So where do we get these high-dimensional vectors? So these vectors are learned from training on billions of words. So here's one, just like very high level, one self-supervision method. You have sentences, you hide a token, you mask it out, and you train a model that tries to predict it based on the surrounding tokens using these deep learning, and in this case, transformer models. So the input here is how are masked doing today, and your target, of course, is how are you doing today. And so as part of this, vectors are learned for each word, and the learned vectors capture what is permitted in the language. So these language models learn the syntax and semantics of human language, as well as protein language. And in particular, these are attention-based mechanisms that uncover specific relationships between tokens. So in a sentence, Mary went to the park for lunch, and she, the important part to understand what she means is to know that Mary is very important for that. In the case of protein sequences, and the words here again are amino acids, if we want to know what that last H or histidine is doing, the important amino acids are the other H and the two Cs, because this is a cis2 his2 zinc finger domain, and the cysteines and histidines coordinate zinc. So very importantly, while no functional information is used to create the vectors, they capture many different protein functional properties in practice. Okay, so there are lots of protein language models at this time. I'm not going to talk about training a protein language model. We're going to leverage existing ones. And so for the work I'll talk about, we use this one particular model from Burkhard-Ross's group that was trained on 14 billion amino acids across millions of sequences. And they end up outputting for each amino acid a 16,000 dimensional vector. Okay. So what we're going to do for each protein sequence that we want to build an alignment for is feed it through their model, get a vector for each amino acid. And the overall intuition of our method is that homologous proteins from different species will have similar amino acid embeddings. So here's an example of three homologous protein sequences from human, mouse, and zebrafish. Each point here is an amino acid. Its vector representation projected onto 2 dimensions. And you see this kind of visually apparent pattern. And so that makes you think that if you overlay homologous proteins from multiple species, this is going to suggest a new strategy for sequence alignment. Namely, you can cluster amino acid representations to find so-called equivalent amino acid positions across sequences. Okay, and this is exactly what we do in our methods called vector clustering MSA or VCMSA. So what we're going to start by doing is generating these amino acid embeddings for all the sequences we want to align. Then for each pair of sequences, we want to compute the similarities between vectors representing the amino acids. And we're going to find reciprocal or bidirectional best hits, or RBHs. So in this case, the K in the first sequence, it's most similar to R in the second sequence, and the R in the second sequence, its closest thing in the first sequence is K. So that's a bidirectional best hit. And we're going to do that for all amino acids, and maybe this is a set of bidirectional best hits that we get. Of course, if we wanted to align these 2 sequences, we know that we have a conflict here with this crossing edge. And so you can clean this up a little by finding maximal non-crossing matchings for pairs of sequences. And the next thing we're going to do now is build a network using these filtered reciprocal best hits. So here we have a node for each amino acid, and we have an edge between 2 amino acids if they're reciprocal best hits as filtered as just described. Okay, so once we have these clusters, we need to still build our alignment, which means we need to order our clusters. So let's say this is your protein sequence. We're going to walk through the protein sequence and see which cluster that each amino acid falls into. So we're just walking through the protein sequence, and you can see which clusters these different amino acids are found in. So of course, we're going to do this for all of our sequences. So in this case, we have 3 sequences. We're walking through the cluster order by walking down the sequence. We're going to take the union of all these paths, and this is going to create a directed graph. And our idea now is we want to figure out what the ordering is. So we want to do a topological sort on this graph. Of course, sometimes you have an error in your clustering. So an incorrect clustering of your amino acid can cause cycles in this graph. And so we use feedback arc set detection to identify and remove these cycles. And once you have that, you can do the topological sort. Okay, so now we have our clusters. We have an ordering across the clusters. And now we can start building our alignment. So for each cluster, we just lay out the amino acids that were found in that, and that sets up columns in the alignment that we're building. And so in this case, example, we have 4 columns that we call guideposts that we have in our multiple sequence alignment. So now these guideposts actually limit where we look in the future for these bidirectional best hits. We don't look across the entire sequence now. We can limit ourselves to within these guideposts. And then we kind of just repeat this process, and I'm skipping a bunch of details, but you would iterate this until things that have similarity are captured in your alignment. So one thing to note is we have no gap penalty at all, because if a column doesn't contain an amino acid from a sequence, it must be a gap. Okay, so now we can think about results. So I'm going to start by saying that these reciprocal best hits, before doing any of these further algorithmics, actually represent aligned columns. So we looked at 562 pairs of sequences. These are aligned. These are gold standard protein pairs from Hamstrad. And for each gold standard alignment, we computed the fraction of aligned columns that are reciprocal best hits. And then what we're plotting here on the left is a histogram where we're showing what number of proteins has a particular proportion of aligned columns that are these reciprocal best hits. And so what you see is that more than 90% of the gold standard alignments have at least 50% of the columns as reciprocal best hits. So these embeddings really do capture something pretty interesting. Okay, so then if you look at these, you can see that the success of the reciprocal best hits is related to sequence similarity, as you might expect. So on the x-axis, we have the gold standard alignment identity. And on the y-axis, we have the proportion of aligned columns in that gold standard that are these reciprocal best hits. And what you can see is as you have higher alignment identity, this tends to go up. But interestingly, even at pretty low identities, for example, 25% sequence identity, you have some cases where you're getting nearly all of the things that are in the alignment as reciprocal best hits. Okay, so this one did not show up here, so I'm sorry, it shows up on my screen, so we'll just skip this. But I wanted to say here was that if you have the same amino acid across columns in the alignment, they actually embed in different places in this 2D representation of the vectors. Okay, so now we can talk about performance. So what we're going to do is we're going to look at 147 alignments from Quant Test 2. For the results I'm showing you today, I'm only looking at 20 sequences per alignment. This is kind of a proof of concept implementation. And we're going to consider a score of alignment quality that corresponds to total column score, which is the percent of columns where the output alignment exactly matches the gold standard alignment. And on the left is what I want you to look at as a schematic for how you look at the results. On the axis on the right, it gives the total column score for our approach VC-MCA. And on the axis on the left is a total column score for whatever method we're comparing to. In purple, that would be the score for one alignment where VC-MSA does better than the other compared method. And I'm also going to tell you the number of alignments where we do better, and the number of alignments the other method does better. Okay, so here are 2 such comparisons. The first is a comparison to muscle. And so you can see that VC-MSA does better in 107 of the cases, muscle does better in 40 of the cases. The second comparison here is to Clustal omega, and in this case, VC-MSA does better in 103 of the cases, and Clustal omega does better on 44 of the cases. We also colored these dots by their sequence identity, and you can see it's a lot of these purple dots that correspond to low sequence identity, where these methods have more trouble, where we have a competitive advantage. So I'm showing just 2 examples here, but we actually compared to 8 different multiple sequence alignment approaches that are given here, and they're actually ordered roughly by the ones we dominate most to the ones that we dominate least. So I wanted to bring your attention to this one point that's sitting over here. Just as an example, this is a case where our total column score is about 60, But in the case of cluster omega, you can see that it has a total column score of nearly 0. And the max score for all other methods is actually not just these, but for all 8 other methods is less than 0.9, whereas we're getting 60. So in some cases, this kind of approach really can dominate what's going on. Oops. OK, so unfortunately, another, oh, no, we got it here. OK, so for very, this is annoying. I had another figure here that didn't show up in the PowerPoint on here. And what this figure showed, I thought it was an important figure, is it showed the comparison when you split things up by sequence identity and looking at the lowest sequence identity where you have less than 20% sequence identity. You can see that our approach really outperforms the other approaches. And this amount of performance improvement decreases as you get to things that are more similar to each other. I'm not sure why it's showing up like this on this screen. So overall, for very low sequence similarity alignments, we get on average 50 to 20% more correct aligned columns than existing methods. So I want to conclude here by saying that we use protein language models to do sequence alignment. We use no gap penalties. We use no substitution matrices. Even though these progressive alignment methods have been kind of the way to go for the last 30+ years, this is an alternative that does something really very different. I'd like to say, I hope I showed you or convinced you that our method, of course still at the proof of concept state, is particularly effective in the regime of low sequence identity, the so-called twilight zone of sequence similarity. And something I didn't show you, but I wanted to point out, is that actually our method is most effective for aligning the structured portions of sequences. And from doing this work, I kind of made me revisit how I think about alignments, and remembering that in the biological case, what we want are equivalent amino acids that share an ancestor, and that might not always be a one-to-one relationship. So less structured regions in a protein may not have functionally analogous regions or individual positions in other sequences. In terms of future work, there's a lot to do. I think we've kind of sketched out the overall outline of the way we're proceeding, but I think there are lots and lots of algorithmic and engineering improvements for speed. I'd like to just finally say that I think protein language models are extremely powerful. I think that in the future, they'll really, in the near future, they really are going to be replacing these substitution matrices, which were a nice approximation of what you could do, but now with including context, I think you can do a whole lot better. So with that, I want to thank you. You can download the software, read the paper, and I'm happy to take questions. Before I do, though, I wanted to make a plug for the Journal of Computational Biology, where I'm editor-in-chief. And this is a journal that's for the community, by the community, and I hope you'll submit your methods-oriented work to JCB.
Thanks.